Right. Good afternoon, everybody. Thank you so much for joining us for today's Leaders Initiative seminar. My name is Stephanie, and I will be monitoring things here on the back end. I will be sending out a Qualtrics evaluation later on, so please take the time to fill out the evaluation. Let us know what topics you're interested in learning about in the future, and we're interested in hearing your feedback on specific topics. Please join us next month. Next month, our Leaders Initiative seminar will be Monday, November 25th from noon to 1. Dr. Pilar Rivero Rios will be presenting mechanisms of altered receptor recycling in Alzheimer's disease. So mark your calendars, and I'll be sending out a calendar invite for that event shortly. And with that, I'll turn things over to Dr. Courtney Polenic to do the introductions.
Great. Thanks so much, Stephanie. And thanks so much to everyone for joining us today. So we're excited today to welcome Dr. Connie Wu as our speaker for this month. Dr. Wu is a research assistant professor in the Life Sciences Institute and an assistant professor in biomedical engineering at the University of Michigan. Her research develops technologies for biomarker signature discovery and RNA therapeutic delivery with applications across cancer and other diseases. So with that, I'll hand it over to Dr. Wu.
All right. Can everyone hear me? All right. So thanks, Courtney, for the introduction, and thanks to Stephanie for setting this up. And thank you, everyone, for attending today. So today I'll be talking about our lab and some previous work, the foundation for the work in our lab now on ultrasensitive multiplexed single molecule detection platforms for a variety of diagnostic applications. And so I realize this is, I'm talking to an audience that's more for Alzheimer's disease. So I'll try to make some connections or potential applications of our technologies to Alzheimer's disease. But in general, so just to give a heads up, the presentation today will focus more on kind of the technology development and also example diagnostic applications. But these are meant to be broadly applicable across a variety of diseases. As Courtney mentioned, our lab focuses on both RNA therapeutics and single molecule detection tools for diagnostic applications. So one half of the lab focuses on engineering what we call multifunctional RNA molecules or RNA systems for applications as cancer vaccines and also as infectious disease vaccines in the future. And then the other half of the lab, which I'll focus more on today, is the development and application of these ultra-sensitive single molecule protein detection platforms for various diagnostic applications so our lab covers, the diagnostic work in our lab covers both technology platform development on developing and expanding such ultrasensitive detection tools to also more clinical application kind of projects that are applying the existing single molecule detection tools to specific biomarker candidates of interest for various disease applications. And so I'll start with this slide where we're mostly interested in, we've been so far mostly interested in proteins. And so proteins are great because they are where the action lies. So they play the direct functional roles in many biological and pathological processes. And so the human genome itself is known to have about 20,000 different genes. But then if you then take into account things like alternative splicing, post-translational modifications, then you actually end up with a whole many more than 20,000 different proteins. So we'll call these broadly proteoforms or different forms of these proteins. And so there's a huge array of human proteoforms that carry a lot of valuable biological information that could be useful for both basic discovery applications and also diagnostic tools serving as potential biomarkers for diseases. And so if you think about the human proteome, much of it is actually, you could think about it as an iceberg, where much of it is actually buried beneath sea level, where at many of these proteins exist at very low levels that are difficult to detect by conventional measurements. And so the part that's sticking up above sea level is actually only a very, very small proportion of the proteins in our proteome. So for example, if you take plasma, plasma consists of many, many different proteins, but if you think about it, so 99% in 99% of the mass in plasma, that 99% is only made of 22 different proteins. So these are super highly abundant proteins such as albumin. But that remaining 1% is actually made up of many, many more proteins, in some cases, estimates of over 10,000 different proteins. And so many of these remain either poorly characterized or in some cases undetectable because they exist at such low abundances and they're masked by the very few but very abundant proteins in blood. And so that's just blood, for example. If you take other biofluids such as urine or saliva, a lot of these proteins are even more diluted in these biofluids. And so that highlights the need to be able to develop sensitive tools to be able to detect such low levels of proteins for various biomarker signature discovery applications, but also fundamental studies. So most of you have probably used ELISA. So the most common protein detection methods are based on enzyme-linked immunosorbent assays. So antibody-based, often sandwich assays where you rely on antibodies to or other affinity reagents to specifically bind your target of interest. So that all works great for many different proteins, but conventional ELISA generally has only low picomolar detection limits, so 10 to the minus 12 molar concentrations of proteins. But there's still a lot of proteins in our blood or in other biofluids that exist below these detection limits, so that remain undetectable by ELISA. And so we really need to have ultra-sensitive tools that can detect below picomolar levels of proteins to be able to better study those many different proteins that are at low abundances in blood or other biological fluids. And so one way to achieve more sensitivity is by going digital. So these are digital measurements. And so just to, as an analogy to show the difference between analog and digital measurements. So the top is what you would normally see in like your regular ELISA. So let's say our readout is fluorescence and optical readout. And so generally, as your target concentration increases, your readout, your fluorescence will proportionally increase in that well, as you see on the top. But the problem is when your concentration becomes too low, or the number of target molecules becomes too low, too few in that volume, the overall concentration of that target molecule in the volume becomes too low to be detectable by conventional instrumentation. And so this is where digital measurements come in. So in digital measurements on the bottom, these measurements basically rely on compartmentalizing single molecules into these ultra small volume containers. So for example, microwells, which is shown here, or water and oil droplets. And so you isolate single molecules in these ultra small volume containers. And so the local concentration of each single molecule in these ultra small volume containers is actually at a high enough local concentration to be detectable by conventional instrumentation. And so by digitizing or compartmentalizing the signal, you can now rely on basically the presence or absence of signal in each compartment and count single molecules one by one versus trying to integrate the signal of many different molecules over a large volume as an analog. And so here, because you're confining single molecules into these ultra small volume containers, now you get much higher resolution at single molecule resolution, and that can help you also achieve higher sensitivity in your measurements. The other nice thing is that you can also better parse out the heterogeneity of different populations. For example, in enzyme populations, there's actually quite a heterogeneous range of activities. And so digital methods also allow you to probe the heterogeneity of different protein populations. So you can apply such digital techniques to protein measurements to ELISA in order to achieve much higher sensitivity. And so digital ELISA methods rely on two main aspects to be able to achieve much higher sensitivities than conventional ELISA. And so the first aspect is a matter of transport or kinetic limitations that exist when you have very low concentrations. So on the left, normally is your conventional plate-based ELISA, when you have your antibody, capture antibody on the plate bottom, and your target protein shown in red here diffuses to a bottom, is captured by your specific capture antibody of interest. And so this all works well at relatively high concentrations, but when you get to super low concentrations, such as femtomolar protein concentrations, so like 10 to the minus 15th molar concentrations, then you become diffusion limited. So you're relying on the passive diffusion of very few target molecules to a 2D surface. And it can take on the order of months even for the system to come to equilibrium. So in contrast, on the other hand, on the right, in digital methods, we use an excess of antibody-coated beads that is well mixed with the solution. So in this case, you're bringing the surface to the molecule instead of waiting for a few molecules to diffuse to a 2D surface. And so you're able to overcome the kinetic limitations here at lower concentrations by using bead-based capture. The second key point for digital ELISA is that we're using a high excess number of beads over the number of target molecules. And so this is the digitization part, where by using a high excess number of beads, you now ensure that the target molecule is distributed over the beads following a Poisson distribution. So basically, since you have so many beads over the number of target molecules, most beads will carry either one molecule or 0 molecules. And so that's the digitization of signal. So most beads actually will carry 0 molecules, and only a few beads will carry 1 molecule. And of course, an even smaller population will carry statistically over 1 molecule. But generally, most beads will have either 1 or 0. And so this has been applied and commercialized in a microwell-based array platform called single molecule arrays, or SIMOA. And so this is probably the most common form of digital ELISA in the field right now, where again, as I mentioned in a previous, I use an excess number of antibody-coated beads. You capture single molecules by and then you can form your single amino complex sandwiches with a second detector antibody. And then you label these single amino complex sandwiches with an enzyme. And then now you load the individual beads into a microwell array. And the key point here is that each well is sized so that it can physically fit at most 1 bead. So in this case, so in this way, you're ensuring that every well has either 1 or 0 molecules. And so you load these beads into the microwell array along with an enzyme substrate that's fluorogenic. And then once you seal the wells with oil, any well that contains a single enzyme molecule on the bead will turn over the fluorogenic enzyme substrate to generate a fluorescent signal. And that's at a high enough local concentration within that very small microwell volume. And that's able to be detected by conventional imaging. And so in this way, you're able to, each well that contains a single enzyme molecule will light up, as you show here. And this way, you're counting the number of molecules by counting the number of fluorescent on wells. And so it's a ratio, the fraction of the on wells over the total number of off beads that gives us our signal readout. And so this method allows us to achieve about 3 orders of magnitude higher sensitivity than conventional ELISA because of this digitization of signal. And also it allows us to use much, much lower concentrations of affinity reagents to lower background signal. So this is all well and good, and it's been commercialized in instrumentation called the HDX analyzer. And so it's actually quite a big instrument, and it's not very well, it's not super well accessible by many labs. And you could, another thing is that this only reaches femtomolar or sub-femtomolar limits of detection. And so there still exist quite a few proteins out there that SIMOA is still not able to detect. So we need to improve both sensitivity and also accessibility and streamlined processing for digital ELISA in order for this technology to be more applicable across many labs and also be able to detect more protein biomarker candidates of interest. So as I mentioned, it achieves femtomolar levels, but there still exist lots of proteins, potential protein biomarkers that Simoa is still not able to detect. And so here in our previous work, we've tried to tackle those challenges that I've mentioned by coming up with a different form of signal amplification from single molecules that allows us to move away from the microwells and use conventional instrumentation and also achieve higher sensitivity, which allows us to access previously inaccessible low abundance biomarkers. And so here, so if you remember previously, we had this really diffusible enzyme substrate that's encapsulated into microwell compartments. And so here we localize the signal amplification to the beads so that this generated signal is attached to each bead. And so the goal here was so that we no longer have to compartmentalize the beads into microwells or droplets. And that allows us much more flexibility in signal readout. And so here, again, we start with an excess number of antibody-coated beads. We capture single molecules and we form our single amino complex sandwiches. But now instead of labeling our single amino complexes with enzyme, we're now labeling with a DNA template. And so this circular DNA template then allows us to carry out a form of isothermal nucleic acid amplification called rolling circle amplification. And so a DNA polymerase can traverse repeatedly around this circular template to generate this very long product, DNA product, consisting of many repeats of that sequence that's encoded by the template. And importantly, this whole DNA product is attached to your amino complex. And so now you no longer have to isolate the beads into your microwells or your droplets, because now the signal is attached instead of freely diffusible. And so this localized signal amplification then allows us to have much more flexible readouts. And so here we chose flow cytometry, which is often used for single cell analysis. So since these are micron-sized beads, we can easily flow the beads through flow cytometer and count the number of fluorescent on and off beads, as shown here. So you can see on the x-axis here is the color of so here we label the DNA product with fluorescently labeled DNA probes that are complementary to each repeat. And so that generates a high local density of probes that's attached to every on bead. And so we can count the number of on and off beads simply very fast by flow cytometry, which is accessible to many different labs. And so we call this molecular on bead signal amplification for individual counting, or mosaic. So this is a very high throughput readout. The other important thing is that it allows us to achieve higher sensitivity than Simoa, because now we're actually able to count many more events, count many more beads and thus events than in microwells, because previously the efficiency of loading beads into microwells is actually pretty low. And so we're actually counting very few molecules, especially at very low concentrations, and that reduces precision and therefore sensitivity. But with flow cytometry, we're a solution-based readout, we can read many more beads and thus many more events, and that improves our precision. So if we compare mosaic and Simoa for a variety of different representative cytokines shown here, in general, we achieve anywhere from 3 to 12-fold increases in sensitivity over Simoa. So these are shown in attomolar. So attomolar is 10 to the minus 18th. So in general, we are able to achieve low to mid attomolar sensitivities across a range of different cytokines. Just to give you an idea here for IL-12 P70, as you see here, we achieved even 5.8 attomolar limits of detection. Just to give you a sense, that's in our typical 100 microliter reaction volume. That's only a few hundred molecules. So that's very few molecules, for protein detection. So that's just to showcase how sensitive we're actually getting. So now, compared to Simoa, we're about an order of magnitude higher sensitivity, compared to conventional ELISA, about 4 orders magnitude higher sensitivity. So here, just the other thing I'll mention is multiplexing. So I've talked about sensitivity, but we're also able to potentially now have expanded multiplexing capabilities with mosaic. So the ability to measure many different protein analytes at once simultaneously in a single measurement. And so previously in digital ELISA, how you would multiplex is that you would have different bead types, fluorescent dye encoded beads, each corresponding to a different target. And so that was all well and good, but you're limited physically limited by the total number of microwells or compartments that you can fit some number of beads in, and also limited loading efficiencies of the beads in microwells. So the highest plex that's been reported to date for Samoa is 6. But with our solution-based readout with flow cytometry and mosaic, in theory, you can flow any number of beads, any number of bead types through flow cytometer. So in theory, you're not really limited as you are in a microwell compartment by the number of different bead types that you can analyze. And so so at least in that aspect, we have expanded multiplexing capabilities where we can do many more bead types. The other possibility, which I'll talk about in the second half of the talk, sorry, I jumped backwards, is that because we use DNA-based amplification, we're potentially now also able to identify RON cross-reactive binding events, which happens in multiplexing when you have lots of different antibodies and targets, and that can generate false positives. So potentially we can use different DNA sequences corresponding with different output fluorescent probe colors, and pair that with different bead types in order to identify RON cross-reactive binding events. And that can better improve that multiplexing accuracy of digital ELISA. All right, so I'll segue briefly into just to show you why. So I've talked about how we're super sensitive, but I also would like to convince you why we want to be super sensitive in some cases. So you might say that many proteins can already be detected by ELISA, but so I'm just going to talk briefly about an example of why how such sensitive techniques have helped us discover potential new biomarkers. This particular biomarker I'm talking about is a multi-cancer biomarker. And so here, this is a collaboration with Kathy Burns and Marty Taylor at Dana-Farber and Massachusetts General Hospital, where we applied single molecule arrays or Samoa and later mosaic to measure this protein that's encoded by this transposable element. And so this transposable element is called line 1 or long interspersed element 1. So it's a retrotransposon. So these transposable elements are what are known as mobile genetic elements, sometimes referred to as junk DNA. In our genome, they make up almost 45% of our human genome, actually. And so these can insert themselves, jump around in our genome, and insert themselves through either a copy-paste mechanism or a cut-and-paste mechanism. In the case of line 1, it's a copy-paste mechanism. And so a lot of these line 1 copies actually in our genome are actually no longer active, but there still remain about 100 intact line 1 copies in each individual. And line 1 is actually the only autonomous transposable element that's still active in humans. So autonomous meaning that line 1 encodes its own protein machinery for retrotransposition. So it encodes only 2 proteins, open reading frame 1 protein, ORF1P, and open reading frame 2 protein, ORF2P. And so just briefly, this is the method by which it copies and pastes itself in the genome. So you have your line 1 RNA that's transcribed, and then it's exported to its cytoplasm. And then it's translated into its 2 proteins, ORF1P and ORF2P. So ORF1P acts as a chaperone protein, a ribonucleotide protein that binds its own RNA. ORF2P plays the more important role in retrotransposition, where ORF2P has both endonuclease activities and reverse transcriptase activities. And so it exports back into the nucleus, where ORF2P can then make a cut in genome and insert itself back into the genome by by target prime reverse transcription. And so you can imagine that you might not want this process to happen normally in healthy tissue because this can contribute to genetic instability. And so normally in healthy tissue, healthy somatic tissue, line 1 expression is repressed because you don't want genomic instability, but it's been found in multiple human cancers, for example, colon and renal cell carcinomas here that I've shown here with IHC. It's actually been found to be de-repressed. So these are IHC staining for ORF1P. So actually in multiple human cancers, actually showing the pie charts here, there's been found, at least by immunohistochemistry, that we have seen de-repressed line 1 ORF1P expression in a variety of tumor tissues. And so the pie charts here represent the percentages, the frequencies in which in each cancer in which ORF1P is detectable by IHC in tumor tissue. So that's in tumor tissue. But then the question is potentially, then our collaborators and I were wondering, hypothesized that maybe potentially by tumor cell death, can ORF1P actually be released into the bloodstream, whether from tumor cell death or extracellular vesicle secretion, whether it could be released in a bloodstream and detected in cancer patients and potentially serve as a cancer biomarker. And so normally it's not expressed in healthy tissue. And so this makes it a very promising specific cancer biomarker compared to many other protein biomarkers. But if it's released in a bloodstream, it's probably diluted and at very low concentrations. And hence we applied Simoa to try to probe the presence of ORF1P in blood. And so we screened a variety of affinity reagents. And so we developed a Simoa assay to measure subfemtomolar levels of ORF1P. We were able to achieve high attomolar, subfemtomolar limits of detection. And so with our first pass, we looked at we tried to apply this assay to measure ORF1P in the plasma of a variety of healthy patients and also different cancer patients. So there's a lot of information in this graph, but I'll highlight the key points. So here, if you look at healthy first, we measured over 400 different healthy individuals. And so the key here, the main point I want to draw your attention to is that ORF1P is super specific. So this red line, dotted line at the bottom is the limit of detection. So anything on that line is undetectable. So we just set it as the LOD. So you can see that in the vast majority of healthy people, ORF1P is not detectable. So this is very, very specific protein cancer biomarker. There are a few up here, and actually this very high one here that's in the red dashed box. Actually, we later looked into clinical records, and interestingly, actually this patient was later found to have prostate cancer. So that was quite interesting scientifically. And now if you look at across different other cancers, you can see, so the pie charts up here across above each cancer show the frequency of in each cancer in which we were able to detect ORF1 in the blood. And so you can see, especially ovarian, colorectal, you were able to detect ORF1P a relatively high number of patients. You can see in varying frequencies among different cancers. And so this can potentially be a multi-cancer biomarker, very, very specific, especially compared to other protein biomarkers, cancer biomarkers, which the problem is they often exist at varying baseline levels among healthy individuals. So the fact that this can be almost a near binary biomarker, it's kind of like black and white, not perfect, but at least much more specific than existing protein cancer biomarkers makes it quite promising. Now, obviously, we're still missing a lot of cancer individuals. Here you can see many of these are still on the red line, and that could be potentially either these patients either don't have line 1D repression in their cancer, and or we could still be more sensitive, and a lot of their levels might be below our current limit of detection. And so we developed an array of different improved affinity reagents to further improve the sensitivity, analytical sensitivity of our assay to see whether we can improve upon the clinical sensitivity of our ORF1P assay in detecting cancer. And so in our second generation ORF1P assays, we were able to achieve higher sensitivity and while also maintaining the same level of specificity. So here's just showing first gen assays on the left and then second generation assays on the right. We were able to detect the red dots show stage 1 ovarian cancer. We were able to detect a few more of those. And if you combine with the ovarian cancer biomarkers that are already existing, CA125 and HE4, so if you only took the existing cancer biomarkers at 98% specificity, you only have 59% sensitivity. But if you combine with ORF1P, you jump up to 91% sensitivity. So ORF1P can be a valuable addition to existing biomarker panels. And just to highlight, to close this part of the talk, just to highlight where our technology development efforts can really further improve efforts in biomarker discovery and improving the diagnostic performance of biomarker assays. So here, all the previous data I've shown you for ORF1 has been just with Simoa using 25 microliters of plasma. And so here, these are 10 healthy and 10 cancer patients with gastroesophageal cancer. All of them were undetectable. And then we applied our second-gen affinity reagents to using Mosaic, which is more sensitive than Simoa. We also increased the volume of the plasma that we use, since there's more, the more volume you use, the more target molecules, especially, which is important when you have a rare target molecule. And so here, by applying Mosaic and larger volume sample volumes, now we're able to differentiate between the healthy and the cancer patients. And so this really just showcases that there's really a lot of value in technology in trying to further improve the analytical sensitivity of our protein detection tools. And that can help us better improve the diagnostic performance for such rare protein biomarkers. And then finally, just to end this part of the talk, so we also showed, besides potential applications in as part of an early detection screening panel, ORF1 can also serve prognostic applications. So here we looked at ORF1 levels in the blood of gastroesophageal cancer patients that were on chemotherapies. And we saw that if patients saw their levels drop, ORF1p levels drop from pre-treatment to during or post-treatment, then generally this was a very accurate predictor of whether the patient would respond to treatment. But if they did not, then the ORF1p levels remained the same or increased. And so this can help us predict potential therapeutic response. And also those cancer patients who started off with higher levels of ORF1 also had lower overall survival. And so potentially, this is just a potential example of how this marker with our ultrasensitive tools could help predict treatment response or overall survival. So that's an example of applying the utility of ultrasensitive protein detection tools, in this case for cancer. We're also interested to see whether we can apply such ultrasensitive tools to measure potential plasma or CSF biomarkers for neurodegenerative diseases, whether it's different forms of tau or other proteins, especially in neurodegenerative diseases where a lot of these proteins in plasma would exist at super low levels. And so now the last part I'm going to talk about our efforts also in multiplexing. So whether in cancer or neurodegenerative diseases or other diseases, often you want to measure a panel of biomarkers, not just one. So it's a single or only a few biomarkers can be difficult. And to improve diagnostic performance, sensitivity, and specificity, you often might want to measure a panel of different biomarkers. But the problem in protein detection with these antibody-based assay or affinity reagent assays is that the more you multiplex, so the more number of targets that you measure at once, the more cross-reactivity you have, and that can limit the accuracy of your measurements. And so by cross-reactivity, it means the incorrect binding of different antibodies to different targets. And so the more targets you have in a multiplex assay, there's an exponentially increasing number of combinatorial interactions between different antibodies and different targets. And that can generate lots of different false positive signals. And that can really reduce the accuracy of your multiplexed assays. So digital ELISA is not, so digital ELISA is also prone, because they're antibody-based assays, it's also prone to such cross-reactive binding and multiplex assays. So here we normally have, so in digital ELISA, we multiplex by using different fluorescent dye-encoded beads for each corresponding to different targets. Then you have your different detector antibodies. And so the problem here is, can you show from dotted lines, there's lots of different combinatorial interactions. This is only a 3-plex here, but if you had like a 10-plex, it would be far, far more. The problem is then if you have a high concentration of, like, let's say one protein, then that can that cross reacts with one bead, then you'll generate you'll generate false positive signal on those beads. And so you're still counting incorrect on beads that result from cross-reactive binding. So your total number of on beads, you have your correct on beads, but then you also have cross-reactive false positive signal from these cross-reactive binding events. So to overcome that, as I alluded earlier, because with our mosaic, we have a DNA, we use a DNA-based readout. So we have a specific DNA template sequence that's attached to every antibody. So now you can potentially use DNA barcoding to identify cross-reactive binding events. So you have, again, you still have your specific fluorescent dye-encoded beads, but now each detector antibody can be barcoded with a different DNA sequence. And that each DNA sequence can then result in a long RCA DNA product that can then be hybridized by different DNA probes that have different colored fluorescent dyes. So basically you can identify only correct pairs, binding pairs, bead color and the probe color, which is dictated by your DNA sequence that's attached to your antibody. And so now you can barcode these detector antibodies so that you count only matched bead, correct bead and antibody pairs that result in on signal. And so this way you can make sure that your signal is only correct matched pairs, and you can remove the wrong pairs from analysis. And so this is what we call barcoded mosaic that allows us to eliminate cross-reactive binding events. So here, just to show again, so normally on the top is our normally conventional multiplexed assay where we just have a single DNA template that corresponds to a single color. And so all your on beads could also include cross-reactive binding events. But in barcoded multiplex mosaic, each detector antibody has a different DNA sequence that corresponds to a different color. And so you'll only count on beads, on events, correct events that are matched bead color and matched output fluorescent color. And so here with the 3-plex, we initially started with 3-plex, so different color beads here as you show here. And so we matched different color beads with different probe colors in each channel. And so only these, so if we only see this bead color with the specific probe color, only then will we count it as true signal. And so that allows us to eliminate all the wrong events from cross-reactive binding. And so here I'm showing you how this barcoded method does indeed reduce cross-reactivity by a lot. So on the top here, we do what we call dropout curves. So we only do single proteins, increasing amounts of single proteins in the 3-plex. And then we look at the calibration, we look at the curves, our signal for each protein in the 3-plex. So we're doing IL-10, 12, and 8 here. And so in non-barcoded on the top here, you can see like when you have high concentrations of one protein, so this is the correct, this orange one here that my cursor is over. So so your on target curve is correct, but then you see the other two curves also start curving up because of the cross-reactive binding. So those are false positive signals that when you have high concentrations of one of the proteins in the multiplex, it can also draw up the signal in the other two off-target beads because of cross-reactive binding. But on the other case, on the bottom now with our DNA barcoded multiplex mosaic, since we're only counting signal that comes from matched bead and matched detector color, output color. Now the other two curves you can see stay flat because even when you have high concentration of the correct protein, the other two curves are not affected by it. So you're not counting false positive signals. And so to further highlight this, so the other thing is that we're able to achieve high, in some cases, even higher sensitivity using barcoded mosaic, in part due to lower background from this method compared to regular mosaic. So in some cases, now we're able to achieve very single-digit low attomolar limits of detection, depending on the antibody pair. And so to highlight, so I've showed you some dropout curves, but to really highlight the ability to improve the accuracy of our multiplex measurements, here we did a range of different mixtures of high and low protein mixtures of spiked proteins, of the 3 proteins in our 3-plex. And so you'll notice, so here we call it the recovery. This is a measurement of the accuracy of our measurements of these different mixtures that have very high concentration of certain proteins and very low of the other one. We compare the accuracies of our barcoded mosaic and our regular non-barcoded mosaic. So here on the left, you can see that especially so the barcoded mosaic is the yellow bars. And so you can see that especially when we have low concentrations of one protein, but high concentrations of the other two proteins, we still are able to maintain pretty high accuracy, so like 100% recovery, which is like a measurement of our accuracy. But in contrast, the non-barcoded multiplex mosaic then achieves much, then gives us much higher actually than actual measurements because it's pulled up by the other two high proteins. So we're able to still maintain high accuracies even in extreme conditions where you have super high concentrations of one protein but low concentrations of another. And we're able to replicate this in plasma as well, where we spiked in different mixtures of these proteins, high and low protein concentrations in plasma. We're able to achieve still reasonable recoveries in plasma, much better compared to non-barcoded multiplex mosaic. So in summary, we're able to achieve much higher accuracies with our barcoded multiplex platform. We're also able, so you might say we're limited by the number of fluorescent probe colors that we can use due to fluorescent spectral overlaps. We're also able to do ratios, have the DNA templates encode different sequences such that we can hybridize different multiple fluorescent probes to the RCA product. And that allows us to, we can use ratios of colors. So instead of just one single color, we can also do ratios of colors to identify as an output signal. And so that further expands our palette of fluorescent output colors that we can use in barcoded mosaic. So here we've done up to 5 plexes, each with a unique fluorescent probe signature with different colors or different ratios of colors. And here we measure a different panel of cytokines and other cancer biomarkers in plasma. Again, achieving low to mid attomolar levels of sensitivity. So this is all well and good in that we're able to now improve the accuracy of multiplexing at these ultra-sensitive levels. But as I alluded earlier, we're still, even though we can do ratios of different fluorescent dyes, we're still, at some point, we will be limited by the total number of fluorescent probe colors outputs that we can use. So our lab is now continuing to try to expand this by using proximity-based barcoding methods, so that we're no longer limited by the total by fluorescent spectral overlap, and we're making sure that the signal is generated only when correct bead and correct detector antibody are matched. And so in this way, we're really trying to achieve higher order multiplexing that is cross-reactivity free. So stay tuned for that. So that's ongoing work. The other thing is that besides, we've talked about a lot of just protein measurements, we're also interested to see whether we can measure other things for multi-parametric profiling. So not just protein concentrations, we're also looking at enzymatic activity, so protease activity, for example, looking at single extracellular vesicles as well, simultaneous profiling of both the markers and internal cargo with our platform. So we're adapting our single molecule platforms to see whether we can measure a range of these kinds of outputs, biomolecules, or their functionality in our platform. So besides just proteins. And of course, we can always try to get more sensitive. Another direction in our lab right now that we're working on is trying to further improve the sensitivity past the even just attomolar limits of detection. So we're limited mostly by, I would say, background non-specific binding. So we're looking at different mechanisms to see whether we can further improve the sensitivity past the attomolar limits of detection in mosaic. And so even with our super sensitive platforms right now, even now, we sometimes are still unable to detect certain proteins in blood or other biofluids. So in these cases, really need to further improve the sensitivity. And so in this case, we're trying to do this by suppressing the background of our assays. And then finally, just to give you another flavor of what's also going on in our lab and in the future. So I've talked about mostly about diagnostic applications, but we're interested to see whether we can apply some of these tools, whether they could be useful for screening RNA or nanoparticle delivery systems. And so here we're looking at trying to do pooled in vivo screening of different RNA nanoparticle formulations in mice, which is normally very difficult to do, not feasible, because it's normally you're just testing one formulation per mouse. But in vitro results often don't correlate with in vivo results. And so you really need to be able to test many different RNA nanoparticle formulations in mice at once, but you also can't feasibly use so many mice. So we're trying to apply barcoded methods to do pooled in vivo screening of our RNA nanoparticle systems. And so these barcodes then can then be read out in the blood or in different tissues with our ultrasensitive multiplex detection platforms so that we can better screen in vivo which RNA systems work well in vivo. And that can help accelerate the RNA kind of drug development pipeline, where that's the goal. So that's also ongoing, so stay tuned. And with that, thank you for your attention. So these are, so a lot of the work I mentioned was from some of the past work and collaborators, which I've listed here. And a lot of ongoing work is now carried out by these members of the lab. And I'd also like to thank you for your attention and funding sources. Thank you.
Great. Thank you, Connie. We can open the floor up for questions right now. Feel free to unmute yourself or place your questions or comments in the chat. I'm going to put up the evaluation into the chat right now. I'll also send it out in a follow-up email after this. If we don't have any questions for Connie, we can end things a few minutes early. I'll just give it another minute. See if anybody has anything to say.
I have more of a big picture question for Connie. You did mention about the intersection between cancer and neurodegeneration. Can you just talk a little bit more about what you think may be going on?
Oh yeah, thanks for your question. I guess I was mentioning more as like cancer, like applications in cancer and also application in neurodegenerative diseases, not necessarily the intersection, though I realized there might be there's definitely also potential intersection. So yeah, I think to clarify, I was talking more about like the broad applicability of our measurement tools for biomarker discovery, whether in cancer or neurodegenerative diseases or potentially fundamental studies, whether in each one or the intersection of those two. So yeah. I would say we're more of a technology development, platform development kind of lab. We're always very happy to, if there are specific potential biomarkers of interest that another lab would be interested in measuring or exploring that could benefit from ultrasensitive tools, we're definitely always very happy to discuss or potentially collaborate.
Okay, if we don't have any other questions for Connie, I will close the meeting for today, and I will see everybody next month. So thank you so much for joining us.
Thank you.